Good morning. Thank you for joining us for our webinar today, and thank you for taking time out of your schedule to be here with us. My name is Dora Taggart. I'm the president here at Microbial Insights, and I'm really excited to be talking to you about our topic today. Many of you have probably heard of metagenomics and next-generation sequencing. And today I really hope to be able to clarify the difference in these tools, as well as show you some of the intriguing analysis that you can do statistically that will help you to make the most of your data. I also hope that I can show you how far you can realistically go with interpretation without jumping to conclusions. So what I'd like to do is start off today by giving you a little introduction, talking briefly about microbial ecology, because really at the heart, metagenomics is a microbial ecology tool. The simplest definition of this would be the study of microbes in their environment and their interactions with each other. For those of you who took any microbial courses in college, you may have heard of Avina Gradsky, who recognized that microbes were the major players in elemental transformations that happen in our soil. Maybe you made a Avina Gradsky column in school like the one that we've shown here, and you can see the stratification of the different microbial groups. You have your aerobes on top, your anaerobes on bottom, and then you have this interaction that's happening in the middle of these two. So this is a really great example of ecology and the direct influence that microbes really have on the big picture of our processes. So with these new tools like next-generation sequencing, microbial ecology is evolving. We're able to understand much more complex systems and complex concepts like the biodiversity that happens in our ecosystems. How stable are these systems? What exchange of growth factors are we seeing? What's the carbon cycling like? And we can start to use these molecular tools to help us understand so much more about our systems. And you've probably seen past webinars that Microbial Insights has done talking about biodegradation. We've got several on our website talking about stable isotope probing, compound-specific isotope analysis. We can use qPCR and quantarray to help us quantify pathways that we're interested in, and we'll talk a little bit about that today in the presentation. But the focus is going to be on understanding what microorganisms are really present in our system. You know, in the past, we used other community profiling tools. Many of you are probably familiar with PLFA. Back in the 1970s, Dr. David White did a lot of work using phospholipids to help us characterize our microbial communities. And we still use this tool today, but what it does is it gives us the 20,000-foot view of what's happening in our microbial communities, which ones are present in our system, but we can't really get genus-level identifications with this tool. So then we had evolution, where we started looking at gradient gel electrophoresis, DGGE type tools that gave us the ability to look at the top 3 to 5 genus that were present in our samples. And we were doing that a lot in the 90s. Then we started doing more TRFLP, where we could look at more genera of organisms that were present. But now with next generation sequencing, we really have the ability to look at much more depth of microorganisms that are present in our sample. And today I want to talk to you about two main types that are used. We're going to talk about metagenomics, and we're going to talk a lot today about amplicon sequencing. And I want to be sure, like I mentioned in the beginning, metagenomics and next-generation sequencing are two terms that really get confused quite often in our industry. So I want to clarify this. If you don't take anything else home, I think this is one of the important concepts. Because although both metagenomics and sequencing are next-generation type tools, they're not interchangeable terms. When we're talking about metagenomics in the strictest sense, we're talking about shotgun sequencing, where we're looking at all the genomes of all the organisms that are present in our samples. So in theory, we're getting the different genes from all the different organisms in our community. So in practice, though, metagenomic shotgun sequencing is rarely performed on complex environmental samples today. From the reports that I've seen, the majority of reports are going to be amplicon sequencing that we'll talk about. because metagenomic sequencing still has a little bit of a way to go because we don't have enough coverage of all these different genes. There are times we just simply don't understand what a gene is or what its function is. We just know that sequence. And then there is a lot of computation that goes into metagenomic type approaches. So we have a lot more cost issues that go into this type of analysis. So what we're going to see, though, as this technology is starting to evolve, even though I think over the next year, you're going to start to see true metagenomic sequencing become more of a common part of our industry. But for now, though, when someone says they're doing next generation sequencing, what you're probably getting 99.9% of the time right now when you submit it for sequencing, you're getting amplicon sequencing. This is where we're looking at the 16S ribosomal RNA gene. So with this, we're amplifying a specific gene, this gene that's found in all bacteria, that's versatile, so we can tell different taxonomic identification of these organisms. So meaning by amplifying this gene and looking at it, we can figure out which microorganism is there and answer that question, who is there? And that can be an important question for us. So before I dive into this a little bit more, let's talk about the workflow, because this is really important for us to make sure that we understand. When we're talking about sequencing and doing the depth of sequencing that we want to do with NGS, we need to make sure that we're gaining quality data. In order to do that, we have to think all the way from sample collection all the way through the process, because high-quality DNA is even more critical here than it is for most of the other molecular tools that we use. So be sure that when you collect your samples, you're keeping them on ice in the field, you're shipping them as quickly as possible to the lab, and make sure the labs are doing the extractions very quickly, because our microbial communities can change over time. So we need to make sure we minimize those changes from field to lab as much as possible, and that we make sure that, because even the small changes in our community when we're doing depth of sequencing can have an impact on what our interpretation can be. Also, be sure that you're using a lab that does the extractions really well. Make sure they have extraction experience in soil and groundwater, because this isn't a trivial process. There's a lot more inhibitors and things that can interfere with getting a good quality DNA extraction. So make sure the lab has the expertise in environmental samples. I personally have a lot of experience extracting medical and environmental, and there's no comparison. It's much harder to get this high quality, good DNA from our environmental samplers. So just be sure that that lab has experience in DNA and RNA from groundwater, soil, and sediments, that they're doing this full time, that that's their job. So when we have our high quality DNA extracts, we can start the process and start sequencing. And again, we're talking typically about looking at the 16S ribosomal RNA gene when we're talking about environmental applications. And this is going to answer the question, who is there? So having the idea of who is there is great, but it can only tell us so much. The real power in sequencing, or any community analysis technique, going all the way back to what we were doing with PLFA, lies in making comparisons. We go beyond just who is there by comparing our results and looking for differences in our microbial community between samples. So we can do this over time. How stable is our community? Is it changing just over time? What does it look like before and after we do a treatment? What was the shift that we created? Did we have the intended shift from our treatment? How does our background look versus source area versus down gradient? So we get a good view of our plume. And then what organisms really thrived in this environment? You know, if we add this selective pressure by doing some type of treatment or having a contaminant, what microorganisms really grew and thrived under those conditions? Who didn't? Who did we get much lower concentrations of these, or much lower abundances of these organisms, or maybe lost them and aren't detecting them at all now? And then who didn't change? And then the big thing that we should always ask is, do these changes make sense? Those of you that know me know I'm a big advocate for having multiple lines of evidence, because we want to see if what we're finding in the microbial community makes sense in light of our site activities. Do the changes in our geochemistry reflect changes in our microbial populations? Do they fit together? And then what I think I'm going to show you today is also other molecular tools can be a real complement to our next generation sequencing so that we can build some much stronger conclusions by having a combination or a tiered approach of these types of tools. So now let's dive into some data and kind of get to the meat of where we're going. For those of you who haven't seen these types of reports, the first thing you're going to get when you do next generation sequencing is this, we're talking about again the amplicon sequencing, looking at our 16S gene, is you're going to get reads. So you're going to get a table like the one that I've shown here where you have your samples listed and then the number of reads that were detected in our sample. You're also going to see the percent of reads that were classified to a level. And for each sample, you're going to get probably a bar graph like I've shown here where it shows the ones that were classified to different levels. And as you can see, there's a drop-off in our species level of what we can classify that far. Usually, kind of the gold standard of where we want to be is looking at a genus level identification. So we're going to be able to look at the percent of reads that were classified to a particular genus in each of our samples. And you can see that here. It's still a really good amount of our sample that reads that were classified. And this is personally where I find a good place that we can look for differences in our microbial community. Now, there may be times, depending on the question that we're trying to answer, that we'll look at these other things like a class or an order. And then, like in the presentation I'm going to show you today, there's value in going down to that species level, even though we can't classify everything, and looking at differences between samples at a species level. Another important tool that you'll find in that table above that we'll mention a couple times today is called the Shannon Diversity Index, and you can research and get more information about this, but it's a good tool for us to use comparatively to help us understand diversity differences between each of our samples. So once we have that table and we know how many reads we've got of these different genera that are being identified, then the question is, who is it? What genus do we have in each sample? So you're going to see a table and a bar chart or a chart similar to what you see here. And what we can do is look at this table and look at the different genera of microorganisms that were classified to a specific genus level. Now, I want to be sure that I point out here when we're talking about our percent of total reads, this is not quantitative. It's used for comparative purposes as an indicator of relative abundance within our sample, but it's not going to be quantitative like we get from a qPCR type approach. The other thing that I want to point out is, and this is very, very important, is you're going to get in this table a description of some noteworthy characteristics of each genus that we find in our sample. And we'll talk about this in a lot more detail during the presentation today. But knowing what an organism is, even at a genus level, can't tell you exactly what they can do most of the time. And you know, you see labs that are trying to overinterpret this, whether it's from an array or from NGS sequencing, and they're trying to assign function to organisms. Sometimes we can do that, like with the Halococoides, but most of the time, the largest majority of the time, we simply cannot do that. And so we can overinterpret our results and really jump to some wrong conclusions because the descriptions in this are generalizations about a genus that are based on what's available in literature. So by generalizations, I mean we're talking about traits that are common among the isolates described. This description often relates to environmental remediation, and we may talk about interesting abilities or phenotypes that will characterize some strains in the genus and species that we've identified. And while these are noteworthy and interesting, they are by no means universal to that genus as a whole. For instance, with Geobacter, we're going to see that a little later in the presentation today. Actually, it's quite the opposite. Some of the things that we may be interested in, those phenotypes that we want to see, are often rare within a genus. So I just want to caution you to be really careful in your interpretation of this. Okay, so we'll talk about that a little bit more later. What I want to do is show you some of the great statistical tools that we have available that we can start to look at these differences. So we've got the reads from our sample. We've got these genus level identifications. What do we do with this? Well, what we found is a good place to start is by looking at something called hierarchical clustering that's included in our report. Now, before I get started on this, I want to freely and happily admit that I am by no means a statistician. I had a few statistics college courses, but I am not an expert in this area. I'm just going to attempt to sum up what we find about these tools, and then, if you have more questions, I'd be happy to try to address them or look up additional information for you. So let's start with this hierarchical clustering. This is an iterative procedure where clusters of samples are created based on similarities between those measured variables. So in our case, a variable is the genus that was detected in our sample. And the value of the variable is that percent of total reads that I showed you in that last table, or that's the relative abundance of that genus that was detected in our reads. So what we end up with when we do this analysis looks like a tree. Samples coming off the same branch are more similar to each other than different branches. So more precisely, the microbial community on the same branch are more similar to each other than microbial communities on different branches. So if we look at the example that I've shown, there are two main clusters. You can see the first pretty easily is MW3, 4, and 6, and the second is MW1 and 2. And these stacked bars below each sample represent the microbial community of each sample at the genus level. So each of those colors that you see in that stacked bar would be a different genus of microorganism. So I think that you can see, just by looking at them qualitatively, that you can see those major clusters. Now if we take a look at that first cluster, we can see that MW3 is on its own, and we have 4 and 6 that are subclusters. So 4 and 6 are more similar to each other than 3, but as a whole, those 3 are more similar than they are to 1 and 2. Now, this is an important side note for those of you that may not be as familiar with this. We have to think of these trees kind of like a child's mobile, and you see those little circles that are the nodes for each branch. We have to be careful to remember that those nodes can rotate. So just because 4 is close to 3 in our example here, it doesn't mean it's more similar to 3 than 6 is, because that could flip and rotate. So we can't just look at those physical characteristics of how physically close they're showing on our tree. So just be sure you're going up to that node level and taking a look at it. Okay, so great, I've got this. What does it really mean to me? Well, we can look for common threads within a cluster, and we find some really interesting similarities and differences based on this. So perhaps, like in this case, our microbial community composition is different because we're looking at impacted versus background samples. And we find this very commonly to be a reasonable situation to see, because the presence of a contaminant can certainly exert a selective pressure on our microbial community. And we can see that some organisms will take advantage of that contaminant and really grow and thrive. Others may prosper under new geochemical conditions. Maybe it became more anaerobic in that area, so we start to see more anaerobic formation. Another common thing that we see that can be differentiating is time. A baseline versus a post-treatment could also cause differences in our microbial community. So you get the idea of what we're finding here. So this is a good quick visual for us to look at how our samples cluster together and group. And then there's a lot of ways that we can move forward with analyzing our sequencing data. Those of you that are statisticians or have more expertise in this, you can start to go in and do more non-metric, multidimensional scaling. You can combine your microbial data with the chemical and geochemical data and start to make some strong correlations that can be very beneficial at your site. But one of the most common approaches that we see in literature, and the thing that I like the best as our second tier, is to do something called the Principal Components Analysis, or a PCA. Again, I'm not a statistician, so I'll summarize it, but you can also go online, and I've put a website here at the bottom of the page that has some really good information at Oklahoma State that kind of defines this a little better. Okay, so with PCA analysis, as I mentioned previously, we're going to take each of our variables, and each genus detected in our sample is a variable. So in one sample alone, we can have 50 to 100 different variables that are detected. And then you think we could have 5, 10, maybe more samples. So it's a lot of variables to try to conceptualize. And I don't know about you, I'm pretty good at being able to visualize 2 variables. I can look at an XY plot, figure that out. I can look in 3 dimensions at a cloud of data and sometimes I can figure that out, but my brain's pretty limited, and when it comes to looking at 50 to 100 variables, I just can't correlate that enough. I can't wrap my head around what all of those means and be able to visually see those differences. And sometimes if I do, maybe I'm being misled visually. So PCA is a technique that we can use to prevent this from happening and help us not be misguided, because it brings out the biggest differences and the strongest patterns in our data set. So if you'll bear with me for just a moment, I want to give you a really simple analogy that a colleague of mine shared with me, and I think it's really valuable. So if you imagine shining a light on a cat, and it's going to cast a shadow on the ground. Based on where the light's coming from, we can gain or lose information about that cat. So if the light's shining straight down like I've done here, do you see the shadow below? It looks more like a blob. We're not getting a lot of information. However, if we change it from a few different directions, we can start to see more information about our cat. We may not be able to completely tell from this that it's a cat, but we're getting more shape. And then from a different direction, look, we can actually start to see that I could identify that this is a cat from that shadow, even see the tail, see the whiskers that are being formed. Well, PCA can do this for a large set of dimensional data. It essentially finds the best way that we can cast a shadow that's going to preserve the most amount of information. And then it continues to do that so that we can get different tiered levels of information. So let's put this into action, and let me show you a PCA biplot. A PCA biplot that I'm showing here is the two-dimensional projection of the samples and the variables. So again, talking about each genus that was identified in our samples. So these axes are our first two principal components. So our points in this case are our samples, and what you see, the samples that are more close together are more similar. And these variables, which are our genus level identifications, are displayed as vectors that extend from the origin with a magnitude and direction, and showing us what's pulling and grouping those samples together. So samples that are near the end where the variables are, for instance, the Dichloromonas and MW6-3, those would have higher concentrations of that particular genus. If you imagine that vector extending the opposite direction all the way down to the end, those would have the lowest abundance of, or lowest amount of reads that we're seeing of that particular genus of microorganisms. So like MW9-4 would have a lower amount of reads for Dichloromonas in that sample. So let's put this into a real-world example. I want you to really see some real-world data. We're going to try to dive into this pretty deep here. So this is a site where it's a petroleum pipeline release that's undergoing monitored natural attenuation. The groundwater from the site's impacted by BTEX, and there's some PAHs there, a little naphthalene that's present. Groundwater samples were periodically obtained from 4 monitoring wells. There were 2 wells that were impacted and 2 non-impacted wells. One non-impacted was a background, and the other was a side gradient well. And to date, we have 4 sampling events that have been performed. Samples were submitted for both the next generation sequencing, again, amplicon sequencing with that 16S ribosomal RNA gene that we've been discussing, and then they also did qPCR to help us quantify genes specific to BTEX biodegradation, since that's the concern here. And I think what you're going to see with this data set is, again, it's a great example of how we can answer all those questions that I just showed you and be able to look at our chemistry and our geochemistry, and then use these molecular tools to really complement each other and make some very strong conclusions. So based on the sampling plan for the site, we can go back to our original questions that we can address with sequencing. From this data set, we can see what are the differences in the microbial community between our background and our impacted samples, which organisms are really thriving under these changes in these conditions, who's been selected in our impacted areas and they're really thriving. How stable are those communities over time, since we have a time series for this? And then are we seeing dramatic changes in background versus impacted areas? And then, of course, how do these all make sense when we talk about our site history, the geochemistry, and then looking at other molecular tools? So I'm not going to get into the table to show you the percent of the reads because it was a pretty high amount of reads that were detected in each sample, and because of time today, I just want to dive into the statistical analysis. So first thing that we do is the hierarchical clustering, and what I've done for this sample here is to take all the time points and look at them as a whole. So we're looking at all 4 wells from all 4 sampling events. And I know it's difficult for you to see. I just want to point out some major trends with this hierarchical analysis. But there are 2 main groups from that first node up there at the top that we're going to divide into. And they separate along this line. So to make it a little easier to see, I'm going to circle those. So if we look, we have a blue box and our red box. If we look in our red box, we can then start to see a second subcluster of our sample that's being detected here. So we can really start digging in. We can look at the samples in the blue versus the red to see what information and key differences that we have on these samples. We can also start to look at our stacked bar graphs and figure out which populations are causing these differences in our samples. So even just by eyeballing it, I can see that that group in green in our red box seems to be a big difference between our red and blue group. And I want to talk about what genus this is a little later in our discussion. So the next thing I want to do after I have this hierarchical clustering analysis and I have these big divisions is to dive into a PCA plot like I showed earlier. And this is actually the same plot that I showed. I've just removed the variables so we can start by concentrating on the samples. So if we just take a quick look at the plot, you can see that there's a group in our upper right quadrant. Then, and that's the blue box. And if that's the same blue box that we would have seen in our hierarchical clustering, and then we have the same red box, which is more of a scattering of samples from the other quadrants. So let's start out and focus on the blue box. And you probably couldn't see it in the hierarchical slide, but this group that I've circled in blue is composed entirely of samples from MW6 and MW7. So what makes this interesting is that these groups are from our non-impacted wells. So that first division that we saw with our PCA plot where it casts the light, the biggest difference we see is between non-impacted and impacted locations. So if we go back to our original question, we can say the microbial communities are different in our impacted areas. The petroleum hydrocarbon, which is probably one of the most important factors influencing our microbial community, applying that selective pressure here as we might expect. So now let's shift our attention to those impacted wells. So if you recall from the hierarchical cluster analysis, we could see 3 subgroups within our impacted wells. Well, the second principal component analysis doesn't show that as clearly, which is why I like to have both of those for comparison, both of these statistical analysis. But what we can do is start to look at our samples in these different quadrants and see what happens. If we look at this upper left quadrant, we see that the first 2 time points, time 1 and 3, are the ones that are grouping together and clustering here. Our second time points are the ones in these lower quadrants, our 4th and 5th events. So it's not a perfect trend, but it certainly appears that over time, or at least during those last two events, we've had a change in our microbial community composition. So that's something that we can start to look at because now we've established some patterns in our data, impacted versus non-impacted, and then early sampling in the impacted wells versus recent. So 2 major differences we want to focus on as we move forward. So before we continue, kind of summarize these differences in our microbial community composition. We see that the microbial communities in the background samples are similar to each other and that they seem to stay very stable over multiple sampling events. And I didn't highlight it, but the values for the Shannon genus level diversity index were lower in our impacted samples, suggesting we had a decrease of diversity, which makes sense once we have a selective pressure that's applied to our community. And as I mentioned, the background wells seem to be pretty stable over time, but the impacted wells had a change that happened. So how were the microbial communities different? So let's dive into that and try to figure out what genus level organisms are causing these differences in our samples. So if we go back to our cluster of samples, the samples that are associated with our background locations had higher relative abundance of Dichloromonas and Methylatinera species than other samples. So also recall that the vectors extend in both directions. So that means they had lower abundances of Geobacter and the Albitoferax, and we'll talk about these more in just a moment. So from our non-impacted locations, what do we know about these microorganisms? Well, the Dichloromonas are generally facultative. They're aerobes, but they can also use nitrate as an electron acceptor if oxygen isn't available. And many isolates have been described to be able to use perchlorate as an electron acceptor. Some strains can degrade BTEX, others cannot. Since these are non-impacted wells, that is probably not likely the case. Methylatinera are methylotropes that are aerobic C1 utilizing organisms. Some can use ethanol for carbon and energy. So as I mentioned earlier, one thing we always want to do with this data is combine it with our other lines of evidence to make sure that it makes sense. And what we do when we see here is that, yeah, the ORP is reasonable. It's mildly aerobic or anoxic, and there's no substantial evidence of iron reduction. Methane is being detected, but I'm getting ready to show you some methane values for our impacted wells, and you're going to see that these are pretty low numbers in comparison. But all in all, the geochemistry looks like these locations would be really good environments for methanotrophs and facultative organisms like the Dichloromonas to survive. So it seems like the sequencing these locations makes a lot of sense. So now let's turn our attention to that red cluster of samples from our impacted wells. From the early sampling events, we see that the relative abundance of Geobacter is higher than detected in other samples. And if you remember, I showed you there was a green bar in that hierarchical analysis that was really coming out that seemed to be the differentiating factor. That green bar was Geobacter. So what can we say about Geobacter? We know that these are iron-reducing bacteria that utilize acetate as a carbon and energy source. Some species and strains in literature have been capable of, shown to be capable of utilizing additional substrates and electron acceptors. Some are even capable of anaerobic BTEX biodegradation. Overall, though, for a genus as a whole, we can say that these are acetate-utilizing iron-reducing bacteria. Now, in our more recent sampling events, we saw that the group of samples was highly relative abundance of Rhodophyrax. And there's been some reclassification in this genus. As many of you know, taxonomy can be more dynamic than we might think it should be because there's frequent reclassifications as organisms are discovered. 16S RNA gene similarities are identified and viewed in different lights based on their phenotypes or other characteristics that may be available. So in this case, the non-phototrophic Rhodophyrax were pulled out, most notably an iron-reducing species, Ferreducin, and reclassified into a new genus, Albitoferax. So the organisms formerly known as Rhodophyrax ferreducens are acetate-utilizing iron-reducing bacteria, similar to what we said for Geobacter. So let's try to figure out in a little bit, why would there have been this shift? Why did we move from Geobacter to this Albitoferax? That was one of the things I thought very interesting in this site. We'll talk about that in just a moment. So from our impacted to non-impacted locations, we can see that the microbial communities are different, and there appears that iron-reducing bacteria are really the winners of the microorganisms that were selected in our impacted area. There's a much greater proportion of these sequences in the impacted than the background locations. So how do these results stack up with our geochemistry? Again, looking at that other line of evidence, we can see that enrichment of IRB is consistent with our geochemistry. Our ORP is lower in our impacted wells, and the electron acceptor data is even more conclusive. We see ferric iron concentrations in the impacted wells are much lower than the background, and there's ferrous iron production. Sulfate concentrations are also lower. So a lot of ferrous iron produced could possibly have precipitated out as ferrous sulfides. And then remember that methane number. Methane concentrations are much higher in these locations, indicating that they're more anaerobic. So ample evidence for iron reduction to be occurring in this location. So seeing an enrichment of IRB really makes sense. Okay, so moving to what I thought was really interesting is what caused this shift during the two recent sampling events from Geobacter being more dominant to the albitoferax. Again, both were detected in all sampling events. We just saw higher proportions of the albitoferax in the later events. And there's a lot of factors that come into play, so we need to really dig deep into the site activities and other lines of evidence. So looking in literature, we can find that albitoferax can often utilize other more energetically favorable electron acceptors like oxygen and nitrate, whereas Geobacter have mechanisms. For instance, they have a superoxide dismutase that they use for survival so they can tolerate a temporary shift to more aerobic conditions. There are also some differences in growth rates, efficiencies, yields, and nutrient limitations between these two different genus of organisms. In general, Geobacter can fix nitrogen, while albitoferax cannot. There are publications that suggest that this organism will outcompete Geobacter when substrate concentrations are low and nitrogen is available, whereas Geobacter have a competitive advantage at higher substrate concentrations when nitrogen is limited. So if we're interested in this shift, we can dig into those lines of evidence to really help us understand what caused that change in our microbial community. So likely the change in BTEX concentrations and nitrogen availability may have been the big factor there. So overall, we can draw some very interesting conclusions from the sequencing data, and I didn't focus on it a lot. I just mentioned it, that there was a decrease in the microbial diversity in our impacted area based on that Shannon Diversity Index. That makes sense because we had that selective pressure from our petroleum hydrocarbon being present. So in general, we gave a selective enrichment of iron-reducing bacteria and other anaerobic microorganisms in our impacted area, very consistent with what we're seeing with our groundwater monitoring data. You've probably noticed that I haven't said a lot about BTEX biodegradation. From the sequencing data, we can make the conclusion that some genera of organisms were identified that have species that are capable of toluene degradation. Really, though, we can't make a conclusion any stronger than this. The truth is that it's almost impossible to evaluate a specific phenotype, like the ability to grow on toluene under anaerobic conditions, based on our phylogeny alone. So let me explain why on this. Geobacter were enriched in our impacted wells, but not all Geobacter can utilize toluene. Here I've compiled a chart of results from literature where different Geobacter isolates were tested for their ability to utilize specific electron donors and acceptors. So to make it easier to read, we've marked the positives and left the negatives blank. What we see is on the electron acceptor side, iron reduction is consistently observed among all Geobacter species and strains. But when we get to sulfur, some can use it and some can't. Similar story for our donors. All utilize acetate as a carbon and energy source. Some utilize hydrogen and ethanol, others can't. Some can utilize toluene, which is very important in our case, and others can't. So the ability to anaerobically grow on toluene is not a universal trait or a defining trait for Geobacter like iron reduction is. So we can't make that broad generalization. So the next thing we might do is, remember I told you we can look at different levels of our sequencing data, so we might dive into the species level and see if that can give us some more clarity on our BTEX biodegradation. So here I've shown a breakdown of the Geobacter species that were identified in a non-impacted sample and a sample from one of the wells in the dissolved plume. As you can see, there isn't a huge difference. Some of them are only 0.5% between the two samples. The only big difference is Geobacter lovelii and the SV strain of lovelii, which unfortunately cannot utilize toluene. So it's difficult to make a strong conclusion about a specific phenotype, like the ability to anaerobically degrade toluene based on phylogeny and taxonomy. In other words, who is there can't tell you always what they can do. So be very careful. Again, if you're getting it from an array or NGS sequencing, don't overinterpret that just because you have Geobacter there, it means you've got anaerobic degradation of petroleum hydrocarbons. I've seen this happen in reports. It's really unfortunate. It's just not something that we can say from that level. So if you'll bear with me, I want to give you kind of a cheesy example that may clarify this a little better. Let's say I'm speeding on my way to work, which actually happened to me recently, and you get pulled over. The officer asks me where I'm going in a hurry, and I say I'm on my way to work and give him my license and registration. So from this information, he can identify that I'm Dora Taggart, and it shows the address where I live in the greater Knoxville area. So that's an observation that we can make. Just like with Geobacter, we can say that the microbial proportion in the groundwater samples from the impacted area had a large amount of Geobacter. Known facts there, right? Acceptable interpretation can be that knowing that I live and work in Knoxville, it's reasonable to interpret that I drive on Interstate 40 and Kingston Pike. For those of you that have been to Knoxville, I-40 is the main highway through it, and Kingston Pike is a very long road that goes through most of the town and has the restaurants and bars and banks and Home Depot and car dealerships. So it's reasonable that if I live in Knoxville, at some point I'm driving on I-40 and Kingston Pike. Similar for Geobacter, we know that they can respire iron, and it's common to every isolate that we've seen in literature. So it's reasonable to say that there's probably a shift to more anaerobic iron-reducing conditions based on the increase in the relative abundance of Geobacter. However, it's easy to go too far. For example, you might assume that because I have a little bit of a southern accent, I grew up in East Tennessee, that maybe I really enjoy country music, and I'm a huge Dolly Parton fan since she's from this area. That, on the other hand, would be going too far. This is a specific preference, and we can't leap to that just by knowing who I am. Some Knoxville residents are certainly country music fans and love Dolly Parton. Others, like me, aren't as big a fan of that type of music. So the same can be said for Geobacter and for anaerobic BTEX biodegradation. It is very specific, and it specifically relates to certain microorganisms, so we can't leap by saying we have a high proportion of Geobacter in our sample to it being able to anaerobically degrade BTEX. Some can do it and some can't. So what we need to do then is use these complementary tools like our quantitative PCR so we can start to draw those more definitive conclusions regarding BTEX biodegradation. So here I've shown qPCR results for these same samples. We have BSSA in blue. BSSA is benzyl succinate synthase, and it encodes for the enzyme responsible for initiating anaerobic BTEX biodegradation of toluene and other alkyl substituted benzenes. Then in red, I've also included ABCA, which is anaerobic benzene carboxylase, and it's one of the few known pathways for anaerobic biodegradation of benzene. We can see that BSSA was above detection limit in only one background sample at one time point. The rest were non-detect. In the dissolved plume, however, we have the opposite story where BSSA was detected in high concentrations in all but one time point. I've also included the estimated percentiles from the MI database. So when I say high concentrations of BSSA genes, I mean it was detected at concentrations greater than detected in 95% of other samples from sites around the world. And while it wasn't detected in all impacted samples, we see that benzene carboxylase gene for anaerobic benzene degradation was detected at monitoring well M9, confirming the presence of organisms that are capable of anaerobically degrading benzene. So hopefully what I've shown you today is that by combining these two analyses and doing this tiered approach, we can make some very strong conclusions. Using next-generation amplicon sequencing, we saw selective pressure in our impacted locations, generating more anaerobic conditions and decreasing the overall microbial diversity. This created an enrichment of iron-reducing bacteria and other anaerobic organisms. Then following up with our qPCR, we could confirm that we had the right species and strains that had the genetic potential to degrade our toluene and benzene. Then we combine this evidence with our lines of evidence, looking at the BTEX concentrations going down over time, looking at the geochemistry changes, and we can use these three lines of evidence to really give us some very strong conclusions about the degradation potential at our site. So we have very strong evidence in this case for toluene biodegrading. So hopefully that example really showed you kind of the depth of what we can do with qPCR and or with next generation sequencing and with diving into this. And there's a lot more that we can do too. But before we end the webinar today, I wanted to give you one other quick example because this isn't just important for petroleum hydrocarbons. I wanted to give you a quick chlorinated compound example. Here I've shown a PCA biplot. And I don't have time to run through the detail like I did, but we can see from the microbial community, if we go through the data, that we can link how our microbial community is being pulled from these 4 different monitoring wells shown here into deep and shallow locations. And we can also see at these locations differences in our contaminant, chlorinated ethenes and chlorinated benzenes. These seem to be the biggest differentiating factors in our location and our contaminant as to causing the differences in these microbial communities. So let's concentrate on the genera that's being detected in the sample. The site was really interested in chlorinated ethenes. So if we look at MW1, 2, and 4 for our chlorinated ethenes in our deep and shallow locations, one thing I always want to look for is, do I have Dihalococoides? But when you look at this plot, although I see a lot of organisms that can be important for portions of reductive dechlorination or for fermentation to get that hydrogen being available, like the Clostridium being there, I don't necessarily see any Dihalococoides. So again, this is where I'd want to pull my raw sequencing results and look to see if Dihalococoides are being detected. So when we did this, we saw that Dihalococoides, there were no sequences identified in MW1 or 4, but MW8S had Dihalococoides that accounted for 0.084% of the reads in our sample. Now, that sounds like a pretty small amount of Dihalococoides to me, and it doesn't sound good. So if I were using NGS alone and basing my decisions for my site strictly on sequencing, I would probably be concerned here, and I wouldn't think reductive dechlorination was very likely. But if we include the qPCR results with this data, we can see that Dihalococoides were detected at concentrations of around 10 to the 4 cells per mil in all 3 samples. This is a significant number because that's our threshold concentration that we're shooting for. In papers like the Liu et al. Paper that talked about this, and then in our experience looking at thousands of groundwater samples from around the world, ethene production is observed in about 80% of samples where Dihalococoides has this 10 to the 4 or greater. And if we look at the geochemistry at the site, we see that the qPCR results are very consistent with our groundwater monitoring data. We have vinyl chloride and ethane that are being formed. So why are we having this apparent discrepancy? It really comes down to coverage. NGS can fail to detect low abundance genera. And I know it's hard to think of 10 to the 4 as low abundance, but we're talking about orders of magnitude here. We're looking at total bacterial concentrations that are much higher. So when we look at the relative abundance of Dihalococoides, we're talking about less than 1% of our microbial community. So unfortunately, that just got missed in our next generation sequencing. And that's just the way it is. When we're casting such a wide net like we do with sequencing, we can miss important, functionally important members of our microbial community because they're in such low abundance. And we find this to be something that happens with the sequencing no matter how hard you try to be good with this tool. With any tool, though, there are limitations, and we just need to understand those limitations so that we can interpret correctly. So I don't want you to think of that as discouraging because every tool has a limitation. Next generation sequencing is a fantastic tool for attempting to link site management to microbial community composition. There's been no tool, our PLFA, DGG, any of these other tools that have come close to delivering the amount and the resolution of information that's provided by sequencing. And once we get to metagenomics, we can see so much more and be able to tie those functions in. And it's just going to be a really incredible and powerful tool. But before you submit your samples for sequencing, just make sure the result you get can help you answer your question. There are times, like when we're doing exploration, that this is an excellent tool. If we want to compare samples, it's the best way to go. On the other hand, if we have a specific microorganism or pathway, we might want to use qPCR or a tiered approach, starting with sequencing and then moving to qPCR. Also, one of the more important points I want to bring out today is what I just mentioned. Metagenomics and amplicon sequencing are not interchangeable terms. Don't be fooled in thinking you're getting all the genes in your samples when you're doing this 16S amplicon sequencing. The results there are taxonomy. And one day soon, as I mentioned, I think even later this year, you're going to see more, or early next year, more availability for this metagenomic type of approaches. So we're much closer to being able to get to function rather than phylogeny. And as we previously saw in these examples, sequencing is just not quantitative. The results are relative abundances within a sample. qPCR is what you want to use for quantitative. Right now, it's the best tool that's out there for that. The results are gene copies per mil. You're talking about absolute quantitation, a specific concentration of an organism. Next generation sequencing also gives you, though, an advantage over qPCR in that you're getting your microbial community as a whole. We can't do that with qPCR. We can't target unknowns or overall community profiling with those types of tools. So if sequencing will answer your question, make sure that you plan your sampling correctly, because it's a powerful tool to look at those differences. Either include a background sample or look at it over time or look at a flow path. Make sure you bring in your history and your geochemical monitoring, and hopefully that petroleum sample gave you a great tool of why these are very complementary in these molecular tools, that we can use all of these to tie together to draw some very strong conclusions about our data. So considering multiple MBTs and tiered approaches can be very wise choices for our sites. Like with any tool, be aware of its limitations. I think there's a tendency, and I've heard this when people have called me, that they think sequencing is going to answer every microbial question that they have. Unfortunately, like any tool, it can't. No analysis can do that standalone. Like we talked about, who is there can't tell us particularly the function. Also, low abundance organisms like our Dihalococoides can be missed with this type of sequencing tool. So before I leave today, I wanted to mention that there are different levels of reports that may be even more important to you than other techniques. There's comprehensive and level 4 reporting that's available. With the comprehensive reporting, you can send in your chemistry, your geochemistry, so the microbiologists here or in any lab, and the statisticians that are in those labs can start to interpret that data in addition to your microbial data and really shed some light on the results. With the level 4 data package, you can get the raw data, so you have your entire list of organisms, not just those top genus. So you can get down to very low levels and really critically analyze your data. And if you're a statistician, that's the way I'd go. I'd want all that raw data so you can go in and do your own statistical analysis from these. So I'd like to leave you today with a couple of online resources. I think our website has some really good information about this. Illumina has really been the leader in the next generation sequencing technology. There's some interesting videos on their website. Remediapedia, which is coming out and getting stronger all the time, has some interesting links on it. And then feel free to give us a call. Our project managers here, Kate and Casey, most of you know them. They're very good and can help you to really understand what's happening with your samples and to kind of walk you through any of the questions that you may have. So again, thank you for taking time out of your busy schedules to be here today. If you can go in and enter any of the questions that you have in the GoToWebinar software, I'll be happy to answer those questions for you, and we can kind of talk through some of the questions. Also, feel free to shoot me an email or Kate and Casey an email, and we can go through and answer any of those things for you. Okay, so let's start. Here's our first question. If there was a case that you had a contaminated soil that had almost no microbial abundance, what can you do to carry on with biodegradation? You know, I haven't really seen that very often. You know, sometimes it may be that there are microorganisms in our samples that, especially from soil, it can be difficult to isolate the DNA from that particular matrix. Like sometimes clay will bind the DNA in the soil and it's hard to get it back out. So I haven't really seen a lot of cases where there is selective pressure. If it is, it's something that's pretty extreme because microorganisms have survival mechanisms where something is going to want to thrive and grow in most areas. So I think it would be important to figure out what is going on at your site, what could possibly cause such a restrictive limitation. You know, even in high temperature environments, we tend to find microorganisms. So could it be high salt concentrations? We've seen a few locations like that where salinity could be very disruptive to the cell membranes and cause you to have less microorganisms that are there. So I would really look at your chemistry and geochemistry and try to figure out what could be causing such selective pressure that you would reduce your microbial abundance in a natural environment. Okay, and there's a question about the metagenomic approaches. When do we think that's going to be available? And I kind of alluded to this a little bit in the presentation. There are more and more sequences that are being identified, and you know, as we start to understand these genes and their functions, that's going to make that tool much more valuable. So I think, you know, within the next year, we're going to see this come online as more mainstream. And then over time, as researchers at different universities start to really identify these microorganisms and to identify these pathways and functions, that's when the real power of that tool is going to come about. There is a question that says, can the low abundance be an artifact of the amplification process? Yes. So two things. With any amplification, you can have where the primer set, the DNA sequences that have been designed that are trying to be unique to all microorganisms just aren't selecting for particular groups of microorganisms. You try to cast a really wide net, maybe you can't always do that. So you can get some biases from the amplification process, which is another reason we'd like to move to the shotgun sequencing, because we eliminate that bias when we're talking about metagenomics. We're just looking at the DNA. We don't have that amplification step in there. But unfortunately for now, yes, there can be some biases that are associated with it, and it can cause us to underestimate bacterial populations, you know, depending on the sequence design. I think most of the time, like in the case of the one before where the organisms are low, if it's not from a microbial ecology standpoint, then it may be a difficult DNA extraction, or you know, it wasn't performed well, or we didn't get high quality enough DNA extracts to be able to perform the analysis. I think that's probably a bigger limitation than the actual amplification in that case. Is there a specific DNA, RNA extraction kit that you prefer? That is, you know, really related to labs and their hands, what they do. There's a lot of good ones on the market. Mobio has a lot of good kits. The problem is I don't know any kit that I take off the shelf that I think does an excellent job of doing the extractions. All of them we've modified here in-house and combined methods and done our own modification processes to make them better kits that can isolate more high quality DNA. So unfortunately, I don't know anything you can just pull off the shelf and use. Okay, the next question, what approach is commonly used to obtain geochemistry data? Is it obtained from literature, or are there common techniques to obtain it from a given limited sample where the geochemistry might be well known? You know, you can pull things from literature, you know, looking at sea level versus, you know, other impacted locations, but unfortunately, I think you need to measure your own geochemistry in those locations. Geochemistry as a whole isn't that expensive to perform. A lot of the field parameters you can do yourself, measuring ORP and, you know, your dissolved oxygen concentrations, pH, those kinds of things in the field, I recommend that you do that for your samples because I don't think I've seen anything in literature that ties directly, and I think there's too much variance that can happen that I wouldn't trust just going to literature for those types of values. Okay, well, it looks like that may be all the questions that we have today. Again, we're happy to answer any questions that you have. We will send out an email this week that will give you the link to the recording of this webinar so that you can watch it again. It'll be also available on the resource section of our website. Feel free to email me if you have additional questions. We'd be glad to answer them or send them out to the audience as a whole if we feel like this will be useful information. But again, thank you for your time, and we look forward to seeing you on our next webinar. Thank you.